So the conclusion is that the deuterium concentration of the metabolic water has a great impact on tumor growth, and the diet can modify and can influence the deuterium concentration. The deconcentration of the different food is very, very different. You cannot be able to regulate the traffic with one light. So if we say that the membrane transport would be responsible and give the green light to the cell to multiply, somewhere must be a red light. And this is in the mitochondria, because everybody every day synthesize about 0.3, 0.4 liter deuterium depleted metabolic water. 2,200 patients drinking DW, we can get a four-fold increase in the median survival time.
Hello and welcome. I'm Sara, and today's guest is Dr. Gabor Somlyai, and we have a feast of science and deuterium today because Gabor is going to go over the 30 years of research that he's done on the effect of deuterium and deuterium depleted water on a variety of different cancers. So there's a huge amount of human data which he's going to show us, but also he talks about different studies he's done in cell culture and in mice in order to understand exactly how deuterium acts as the sort of on-off switch or the binary code in what makes a tumor cell grow or not grow. In the second part of the talk, Gabor talks about blood sugar and diabetes and his findings using deuterium depletion. The science and the data that Gabor presents is very strong, yet he goes into some of the reasons why it's so difficult to get new therapies registered for cancer. I've made a Substack of this presentation that Gabor did so that you can follow along with the transcript and the slides, and the link to my Substack is in the description. Welcome back to the channel, Gabor. It's going to be a great treat today because we're going to have one of your presentations all about deuterium and not just cancer, but a really exciting metabolic study on blood sugar. So do you want to introduce yourself quickly if people don't know who you are, because this is your second time on the channel?
Yeah, hi Sara, it's very pleased to see you again. So my background is molecular biology. When I was a kid, I was facing with a cancer because one of our neighbors had that type of disease, and after that at that time I decided to think about cancer. So finally I graduated as a molecular biologist, and I was so lucky that an idea came to my mind, what I started to test 30 years ago at the National Institute of Oncology. And the very, very first experiment proved that my hypothesis is very promising. So in the last 30 years, we completed thousands of experiments and involving lots of other scientists. And for today, we have a very clear picture. What is the reason of cancer and what should be the way to cure cancer?
Okay, so let's begin.
Yeah, so the first thing we are facing that how complex is the living organism. And for an average people who are not molecular biologists, they should think about a couple of data or numbers. So one single cell size is 40 micrometer. Within there is a 10 micrometer size of nucleus. And that nucleus contains physically 1.8 meter length DNA, which contains 3.2 billion base pairs. So this is the DNA which is packaged in a very small nucleus, harboring and covering the 25,000 genes. And each cell's working perfectly. Within that cell, there are 2,000 different biochemical processes. They are harmonized. They know the cells when to induce the expression of given genes, when to suppress. So everything is fine. The question is, what is the problem when we talk about cancer? And the other question is, what is the signal for the cell to start the cell division? And if we go to the next slide, we know that there are 4 phases of cell cycle. typically the cells are staying in the G1 phase. And everybody looking for the signal how and when decide the cells to move from the G1 phase to the S phase. And as you see, it takes about 24 hours to take one cell cycle. And the most difficult question, when the cells decide to move to the G1 phase to the S phase, how can the cell manage the next 10, 12 hours from the decision point? Because the cell never will start from G1 to the S phase if the cells are not 100% sure that they can complete the whole cycle. So these are the big questions we have to answer. And the next slide. And the reason that the cancer problems has not been solved, I think the main reason is that everybody is thinking on a molecular level. The pharma company and the research is working on to find molecule, hoping that one molecule may be binding to a protein or maybe changing or modifying the expression of one gene that can help us to have a good cancer drug. And if you see the numbers, the incidence of cancer is increasing, the number of people dying of cancer increasing, and the prognosis is very bad. So we say that we can change that process, and the future, if we think about the molecular level, will not give us a right answer. And the reason is there is a submolecular regulatory level. And this is what I would say I discovered, I hoped it is there. And these are, we can say 100% sure that the science has proved, involving hundreds of scientists, that the deuterium hydrogen ratio is the key. And if we go further, what is deuterium? Deuterium is a heavy hydrogen. So when we drink a glass of water, we drink H2O, HDO, or DTO molecules. And this can be important because the 100% mass difference between the hydrogen and deuterium that cause a big differences in the chemical reaction when depending on whether a hydrogen or deuterium take part in a chemical reaction. It is also very interesting that the ratio between the hydrogen and deuterium is 6,600 to 1. So everybody can say, okay, let's ignore it because the deuterium level is so rare. But this is not true. This 150 ppm means that in our body, the D concentration is 6 times higher than the calcium level in the blood. So how is it possible that we ignore that? But that was because the deuterium was discovered in the early 30s, last century, and till 1990, nobody investigated the possible role of the deuterium in the living organism. So the question is, the next slide, so in the first 60 years after the discovery of the deuterium, everybody used the heavy water in high concentration. And the slide shows when the tobacco was watering with an increasing D concentration, D2O concentration, the plants couldn't grow, which shows that for the living organism, the deuterium and the hydrogen is considered as two different biochemical components or elements. And all the H bond breaks much faster. And this is the isotopic effect. So when we modify the DH ratio, we modify all the biochemical reactions where hydrogen or deuterium take part. So the next slide, please. This shows the deuterium concentration in our body. And it's clear this shows that how relevant the deuterium concentration in the living organism. So again, the question is, how could we ignore that element, the deuterium? The next, please. So the very first paper we published in 1993, and the title of the paper was Nature Occurring Deuterium is Essential for the Normal Growth Rate of Cells. So in that paper, the next slide shows that we checked the cell growth in vitro. So we prepared media with deuterium depleted media, normal media, 150 ppm, and 300 and 600 ppm. And as the left side of the slide shows, the deuterium depleted media, the cell growth was inhibited, but the deuterium enriched media stimulated the cell growth. So that was published 31 years ago. And in that paper, we also published that when we transplanted human breast cancer to mice, and one group of the mice, the control group, consumed normal water, the treated group consumed deuterium depleted water. And here we found that 11 mice out of 17 were still alive, and they were happy 3 months later. While in the control group, only one mice survived the 87 days, which showed clearly that the low D level can prevent the growth of cancer and can save the life of the mice in that study. So that was very, very promising. And after several years, the technology developed. So we used a so-called X-Celligence system. And the good part of this technology that we do not need to manipulate with the cells. The cells are growing on a gold layer, and we follow how the impedance of this gold layer is changing. And it means the faster the cell can grow, the faster the impedance of the gold layer is changing. And that allows us to carry out extremely precise experiment. Here, we prepared media not only with 150 ppm, that with a lowering 135 and go down to 40 ppm. And again, we could see that the lower the D level, the lower the growth rate of the cells. And with that technology also could prove that increasing the D level over 150 ppm, that stimulated the cell growth. So that way we could repeat the experiment what we published 31 years ago. And the next slide, it's very critical. The question is how sensitive the cells from the changing D concentration. So in that study, we modified the D concentration with 0 ppm, 1 ppm, 2.5 and 5 ppm every 8 hours. And here we can see that the green line is different from the red line, which means that the cells recognize the 1 ppm decrease of the D level, and that resulted a lower growth rate. So it is so sensitive, the system. It means somebody drinking 1.5 liter deuterium depleted water with 105 ppm in a body, we can get a 1 ppm decrease. So let's see the next slide. So in our very first paper, what we published, it was clear, and we say what is the mechanism we suggested. So even in the 80s, everybody agrees that when there is a pH changes in the cell, that can trigger the cell division. So when a growth hormone binds to the membrane receptor, and that stimulates the sodium-hydrogen exchanger, that means the cells pick up one sodium and transport one hydrogen ion. And everybody knows that this is somehow a signal for the cell. But the real surprise was that a paper published in the Nature proved that the hydrogen ATPase gene was isolated from the yeast. And that gene was transplanted to the cell line. And in that paper, proved that the hydrogen ATPase was sitting in the membrane of the cell, transporting the hydrogen ion. But nobody understood how is it possible that cell line caused tumor in nude mice. So we put one gene into the cell, and that make it tumorous. So our concept was when the hydrogen transport system is activated, that will be discriminate between hydrogen and deuterium and only transport the hydrogen. That results in increasing DH ratio in the cell. And we suggested that this is a final signal for the cell to initiate the cell division. So when we use deuterium depleted water in the tissue culture or in the mice study, we reduce the D level and the cells are not able to lift up the DH ratio to the threshold. And this is the reason that we can cure cancer, and this is the reason we do not allow the tumor to grow in the mice. Everybody knows, and Albert Szent-Györgyi suggested, that you cannot be able to regulate the traffic with one light. So if we say that the membrane transport would be responsible and give the green light to the cell to multiply, somewhere must be a red light. And this is in the mitochondria. Because everybody every day synthesize about 0.3, 0.4 liter deuterium depleted metabolic water. It means when we eat the food and we burn the food to carbon dioxide and metabolic water in the mitochondria, that is deuterium depleted. The question is how depleted, and if we see from 100 fat, the mitochondria synthesize 107 grams H2O, from 100 carbohydrate 56 grams, or protein 40 grams water will appear. But if we go to the next slide, we see that the deconcentration of the different food is very, very different. It means eating carbohydrate, our mitochondria will produce metabolic water with 150 ppm or 146 ppm. But the more fat we eat, the lower will be the deconcentration of the metabolic water. So the final deconcentration of the metabolic water depends on the composition of the diet. And I would say the main reason that today there are 400 people with metabolic disease and diabetes and 20 million new cancer cases, that in the last 60, 70 years, the people covering the calorie intake mainly with the carbohydrate. And the fat, it is said this is a bad and wrong type of food. So the average deconcentration is higher of the society which is living in the last 50, 60 years. So in a basic situation is in a healthy cells, the membrane transport can increase the DH ratio, but in a healthy cells, the mitochondria working properly, producing deuterium depleted metabolic water. So the DH ratio is low. But everybody knows since Warburg that the only common feature in each cancer cell is the damaged mitochondria, which means that the cancer cells are cancer cells because the mitochondria is not able to produce deuterium depleted water because the TCA cycle doesn't work properly. So when a growth hormone binds to the receptor, the DH ratio is originally higher, and from that higher level, much easily can lift up the DH ratio to the threshold. And this is the reason that the cancer cells more frequently can reach the DH ratio and more frequently can initiate the cell growth, and they grow. That's it. So the conclusion is that the deuterium concentration of the metabolic water has a great impact on tumor growth, and the diet can modify and can influence the deuterium concentration. The next slide.
I just wanted to ask a question here. So this is really interesting because with protein, we always talk about carbohydrates and fats when we talk about deuterium, but then not a lot of people talk about the consequence of protein being metabolized. Would you just be able to go over that briefly before we move on? Because it made a lot less water than I thought. I thought it was somewhere in between carbohydrates and fats, but I was mistaken. So what happens with protein and deuterium, and how much deuterium depleted water we make?
Yeah, if you go back a couple of slides here, so you can see that the cottage cheese D level, we can consider it as a protein source, is much lower than the carbohydrate, but higher than the butter or lard or goes fat. The reason is the more complex is the molecule, the lower the deconcentration. So the fats is a very long chain carbon source, and during that any chemical step, always the hydrogen will be preferred to move forward with the chemical reaction, and the longer the chemical reaction, the lower will be the D level. And this is between the protein and the amino acids level. Deuterium depletion is between the carbohydrate and the fats, and yeah. So I would say, yeah.
Is there any consequence with deuterium and cancer from consuming too much protein?
I would say the key message is, I say, the ratio between the carbohydrate and fat. Of course, comparing so being on a protein diet, that will lower the D level of the metabolic water comparing being on a carbohydrate diet. But comparing the protein diet with a ketogenic diet, that results a higher D level with the ketogenic diet. So, and I suppose that nobody can eat only carbohydrate or only protein or fat. So this is somehow the combination of these three basic food type.
Okay, yeah, I just wanted to clarify that. So basically the take-home message is it's the carbohydrates and the seed oils, of course, and then the protein and the fat is the ideal way to move forward for lowering deuterium in the diet.
Yeah, this is what we say. Yeah. So if so we know that the science is great. The molecular biology has discovered all the molecular background of the cell division. And we know that hundreds of genes are involved in the cell division. So the question is, how can we modify the gene expression by changing the deconcentration? So that slide, I try to show that we made an experiment when we tested kinase genes and cancer-related genes. And finally, we chose 124 cancer-related genes, where we so that the changes of the gene expression was higher than 30%. So in that study, we kept the cells and deuterium depleted media, normal media, and deuterium enriched media. And the data was very, very surprising that most of the genes responded to the deuterium enriched media, which means a higher DH ratio triggered the expression, not only one gene, but hundreds of gene expression will increase, which is related with the cancer. And if you go to the next slide, which shows the kinase genes. So in that case, we found only one gene which was downregulated in deuterium depleted media, but 134 out of 135 expression was increased in deuterium enriched media. And the point is that, again, the pharma industry is targeting one tyrosine kinase gene, and they hope that one molecule will inhibit one tyrosine kinase gene. Changing the D level, we simultaneously can inhibit the expression of hundreds of tyrosine kinase gene. And this is the reason that drinking deuterium depleted water, that can be effective for all cancer type, and it does, because we simultaneously will intervene into the not only one point of the signal system, but at each point of the signal system. So when we tested a deuterium depletion in a clinical study, that was a very first study with a prostate cancer. We evaluated 44 cancer patients, 22 consumed DW, 22 normal water, and everybody received the hormone therapy. So the treated arm consumed 85 ppm, the placebo 150 ppm. And after 4 months, we found that there was 80% decrease in the PSA volume in the treated arm. That was only 47 in the placebo arm. And it was when we checked the prostate volume, the cumulative decrease in the prostate volume in the treated arm, it was 160 cube centimeter, and that was only 54 cube centimeter. And the most promising result was when we checked the survival after a year, we found that we lost 2 people out of 22 in the treated arm, but 9 out of the placebo, and 9 out of 22 in the placebo arm, which was clear that those group of people drinking DW, they live longer. We could reduce the prostate volume, the tumor size, and that was very, very effective. There are 3 types of products on the market available. First of all, we registered deuterium depleted water as an anti-cancer drug for veterinary use. So that was registered in 1999. So today we can say that dogs and cats can be cured roughly 50% just by replacing the normal drinking water with deuterium depleted water. Our main product is a simple drinking water. This is a food product, so we cannot claim anything to that type of product because this is a drinking water. But most of the study and the results we collected coming from those people who were drinking deuterium depleted water, receiving the conventional therapy, and we got the feedback of the data. And the third is a deuterium depleted mushroom capsule, which is a new way to reduce the deconcentration in the mitochondria, because that mushroom was cultivated on a deuterium depleted water, and that way we can modify the DH ratio in the organic molecules, mainly in the carbohydrates. So when we collected and followed cancer patients drinking deuterium depleted water, we evaluated 2,222 cancer patients. That was evaluated 5 years ago. So the cumulative follow-up time from the diagnosis over 8,000 years. And the bad news is that most of the people come and started to consume DW relatively long time after the diagnosis. It means that most of the people were in a stage 4 and in the late stage. But anyway, the people consume DW for more than 2,400 years, and we could follow them after that over 4,800 years. And checking the median survival time of this big patient population, we found that.
Can I just pause a minute? Because I think when I looked through these slides before, when you're expressing these 2,000 years and 4,000 years, it might be a bit confusing for somebody who doesn't understand what the data is saying.
So it means if we talk about these 2,222 patients, and we see that the cumulative time of the DW consumption is 2,400 years, it means the average, everybody consumed longer than 1 year the DW. If we talk about the cumulative follow-up time, 8,300 years, we can say that typically 4 years. We followed everybody for 4 years after the diagnosis. So, of course, this is not true because there was people who started immediately the consumption of DW right after the diagnosis, and there are people have been following them for 20 years, and they are fine. So these are, of course, rough numbers, but the key message is that based on over 2,000 cancer patients, and it's a very, very long follow-up time.
Oh, no, that makes sense, because then the 8,000 years means people use the deuterium depleted water for 4 years, and then 2,000 means they started to use it later and you were following them for a year. So that just clarifies, because this is now really important data, because these are all these different tumor types.
Yeah. And this is the next slide, which shows what is the distribution in that population, which is roughly cover the same, that the digestive system, breast and lung cancer, they cover roughly 50% of the total cancer population, and depending on the others type, we have some of them very few, and depending on. So, and if we go and we see the number, so what we can follow, which is valid. The median survival time is a number which shows, let's say the lung cancer median survival time worldwide is about 8 to 12 months. That means if we take 100 lung cancer patients, typically within 8 to 12 months, there are only 50 lung cancer patients alive. It means 50% die within 8 to 12 months. If we check the median survival time of the Hungarian population, in Hungary we have 77,000 new cancer cases, and we lost 33,000 people every year. In Hungary, the median survival time of the whole population is about 2.5 years. And that 2.3 years, comparing with the 11.1 year, means that in that 2,200 patients drinking DW, we can get a 4-fold increase in the median survival time. And from that number, we would conclude that in Hungary, we will be able to save the life of 25,000 people every year when the DW will be part of the oncotherapy. I don't know when will that happen. We have been working on for 30 years, and we have not been able to get a registered cancer drug, but this is the aim, what we have to get. To get a cancer drug, integrate to the oncotherapy, and that way get a real breakthrough and reducing the deaths caused by cancer.
Okay, because that's really important, that number, the 11 years, because it's so significantly larger than the number that you mentioned. Because did you say it was on average 2 years or so that people would survive in Hungary? Yet with that data, you're saying that still nobody is taking these numbers seriously enough.
Yeah, this is another story. So how to register a cancer drug, what is the cost to register a cancer drug, and how difficult to convince the society that we should think about another level on a submolecular level. But the good news is that the science already accepted this data. So just recently, 2 review papers was published by Chinese scientists. They summarized the last 30 years of that type of research. So I'm not worried about it. So that will go further, and then more and more people recognize it, and one day we reach it.
Oh yeah, yes, you will, because the data is really strong. So shall we move into breast cancer now?
Yeah.
I want to thank my sponsor, Lightwater Scientific, who helped me deliver free, valuable information to you and also allow me to have great guests on this channel. Lightwater Scientific produce the purest and most deuterium-depleted water in the world. Depleting or lowering your deuterium has numerous health benefits, from better metabolic health, more energy, greater resilience, less brain fog, increased physical performance, enhanced healing, and healthier mitochondria. Deuterium is an isotope of hydrogen, also known as heavy water, because it's bigger and heavier than regular hydrogen. Even though deuterium makes up only 0.02% of hydrogen, it's found everywhere. Deuterium is a burden to your body because it interferes with the intricate chemical reactions that occur by slowing them down. Deuterium is kryptonite to your mitochondria, and we need healthy mitochondria to produce energy. There's over 60 years of scientific research on the negative effects of too much deuterium on health, and also the positive benefits of lowering deuterium in the body. Light water, deuterium depleted water, is 100% safe and non-toxic. In fact, it's the closest in structure to the metabolic water that your mitochondria produce. Light water, deuterium depleted water, comes in 5 or 10 parts per million glass or plastic bottles. So level up your health by leveling down your deuterium today. The purchase link is in the show notes, and use code Dr. Sara for a discount off your order.
So in that case, we evaluated 232 breast cancer patients, and we published it so people can find it and see the data, so people can see it here. The DW, yeah, go. So in that case, we made, so what is the basic numbers? So the cancer patients who are diagnosed in a advanced stage, the median survival time is 1-2 years. Those who are diagnosed in an early stage, the median survival time 6.6, 8.1 year. So, and when we checked our breast cancer population for the whole 232 patients, we calculated 12.3 years, which in general we can say it's a big increase in the median survival time. But then we made different subgroups. So in our population, even at the date of the diagnosis, 74 people were in a stage 4, and 158 were in a, let's say, in an early stage. And if we see the median survival time, so we increased up 4.3 years the median survival time of the people being in a late stage, and the early stage it was 18 years, which is a very good number based upon the over 100 breast cancer patients. And the dosage is very critical when we talk about a drug. So we calculated and figured out a number. If we see these numbers, so if somebody drinking with a body weight 60 kilogram consume 1.6 liter water with 65 ppm, the DD dosage number will be 2.3. But if the body weight is higher, 80 kilogram, they consume less, 1.3 liter of water and only 105 ppm, this number is 0.7. So what will be the results when the people drinking DW in a different dose? So in that study, we followed that 74 breast cancer patients who had metastasis in 135 organs. So some of them had metastasis in 2-3 organs. And the next slide shows. So we made 2 groups of patients, those who consumed DW where the DD unit was bigger than 1, and those where the DD unit was smaller than 1. And in that case, we can see that 30% we can get a complete regression. It means that the tumor disappeared. And in 40%, there was a shrinkage in the tumor. So when the dosage reached the DD unit bigger than 1, then 70% of the cancer population improved. And when it was lower than 1, then they do not changes in the tumor volume, or it was a progression of disease, which clearly shows that how important to adjust the dosage to the given people and the given cancer.
Okay, that's really important, because some people who want to use deuterium depleted water are not going to be working with a doctor or a practitioner. So I think what you've just said about the dose varying from a function of the person's body weight, it's really important that they consult your book on deuterium depletion and cancer, because some people will just watch a podcast or just drink the water neat. So I think this is really important to say that the dosage matters in this case and a protocol is needed.
Yeah, yeah, and of course the best would be when that would be combined with the conventional therapy. But I hope one day that will happen.
Yeah.
So some similar results we published in the glioblastoma, which is a poor prognosis. So here we evaluated 55 cancer patients with glioblastoma, and the median survival time for the whole population was 30 months, which is a double comparing with the historical control. But it is, again, very important. So in one group of the people missed the DW, it means they were operated, they received the radiotherapy, and several weeks or months later started to consume DW. So in that subgroup of the population, the median survival time was 25 months. But those lucky cancer patients, when we received the radiotherapy at the same time they could consume DW, then the median survival time increased up to 47 months, which is extremely big increase in the median survival time in that type of cancer.
Because that's really important as well, because this is showing if you interrupt the deuterium depletion because these people stop taking the water and then start again, it affects the outcome, because the ones who had the 47 months of increased lifespan, they used their DDW continuously in conjunction with their treatment, which was radiotherapy. So that's, I think, is quite an important.
Yeah, and the other message is that the most significant and synergistic effect we can see between the radiotherapy and DDW. So this is the best way when somebody receiving the radiotherapy and at the same time drinking DDW, because both treatment is generating radicals, and that way we can reach the increase of the radicals which can trigger the tumor. So that is the key, that this combination could be so effective.
Oh, right. Okay. Because when I was looking over, I misread this slide. This is why I brought it back up. So I think this is even more evidence for if it's combined with a traditional therapy, the amount of killing or removal of cancer cells going to be greatly increased. For example, with radiotherapy and deuterium depleted water is a very powerful therapy, is what this is actually saying.
Yeah, this is one. Yeah.
Yeah.
So this is the lung cancer. Here I only summarize that it's very interesting that there is a very, very big difference in the response rate between the male and female. And the females over respond much better for the DDW than the males. So in our case, the median survival time of the lung cancer in females from 12 months increased up to over 100 months, while for males we increased up to 41 months. We had some preclinical data what should be the reason that the male respond much better for DW, but it needs more research to clarify. But anyway, looking the lung cancer, it again, we can multiply the median survival time. And this is another very important, it's a Poisson correlation, yeah, which shows if we, okay.
All right. No, I was just going to pause again here, because this is really important, because it's almost, about 9 times more survival in the females. And then for the males, it's about 4 times. And lung cancers are pretty aggressive. So this is huge if somebody can get 9 times more lifespan from it. And also, I think when we did the other podcast, you also brought up this interesting information about the females and the males being different in how they respond to DDW with lung cancer. So is there any, have you had any more thoughts on the mechanism, or is that beyond the scope of this discussion?
So the data we have published, that we checked the expression of a couple of genes when we trigger the mice with a carcinogen agent, and the c-MYC gene was one of them, which was very, very strongly induced in the females. And when giving DW to the mice, we could suppress it. But that gene wasn't so active in the male mice. So we suppose that maybe that should be one reason that those genes which responding and has a very important part in the cell growth in the females is more active, and they respond quite well with the DW, but not so active in the male. That should be one reason, but need more research. And these data shows that the longer the people consume, the longer they live. And that shows for prostate, lung, pancreatic cancer, breast cancer, and colorectal cancer. So the Pearson correlation, not Poisson, Pearson correlation is clearly shows that there is a clear line which shows the strong correlation between the length of the DW consumption and the length of the survival.
And that's, I just noticed, because there's pancreatic cancer here, and that's a very, very aggressive cancer. Yeah. So that of all of the data sets here, we would expect the pancreatic ones to respond the least to the deuterium depleted water. But how much extra life did the pancreatic cancer people get?
So the median survival time could be between 6 to 8 months. It's very aggressive. And these data also show that how important that the deuterium depletion should be part of the therapy, because we need all type of treatment which is available. And it's very important to do more research to even how can we increase the efficacy of DW, what would be the best combination. And so we cannot be satisfied with the pancreatic cancer survival of 1.8 years.
Okay, yeah, because that's another important thing you've brought up, because there are other cancer treatments which also produce free radicals, some of which are licensed and some of which people get themselves. So I think this idea that if you stack deuterium depleted water with other treatments that produce radicals, you're going to get an even better effect, and the deuterium depleted water is not going to impact any other aspect of this person's life. It's actually going to improve their quality of life while they're receiving these treatments. Is that also correct?
Yeah, and maybe the most important, so sometimes I didn't understand why people do not show any improvement. And later it's turned out those who are taking antioxidant combining with DW, that can diminish the efficacy of DW. So we strongly recommend drinking DW, do not take antioxidant, because that way the antioxidants will catch the radicals and will help the cancer cells to survive the treatment of deuterium depletion. And
yeah, that's really important, because this idea that free radicals are just bad is terrible. And I think this is really important for people, because they'll try sort of juicing or buying very expensive supplements that touted as antioxidants, and they're basically making the problem worse. So I think that's a very important point to bring up as well.
Yeah, so it is not true the more type of additional and complementary therapy they try, the results is better. No, it must focus on one or two alternative therapy plus receiving the conventional therapy. That is the way to get the best way, the best results. So the next slide, I would say, if you go back, maybe this is one of the most important, that out of the over 2,000 cancer patients, we had 204 who had been diagnosed, and they were tumor-free when they started to consume DDW. And we know that luckily a big number of the patients can be tumor-free, because the operation, chemotherapy, radiotherapy can be tumor-free. But we also know that, unfortunately, most of them relapse within 1, 2, 3, a couple of years. So we follow that over 200 cancer patients. The cumulative time is over 1,000 years, which means the average time was 5 years every patient. And this is what we found, that 159 patients never relapsed, and we lost only 2 people out of these altogether out of these 159. And these died 20 years after the tumor was diagnosed. And the last line, there are 22 patients who relapsed after finishing the DW. Can we go back?
Oh, sorry.
No problem. And we lost 8 out of these 22. So these people consumed DW, then they finished it, and they never came back. And the next slide shows what is the difference between those those two, or I would say those 159 and the last 22, because who never relapsed, they consume DW, has a break, consume again DW, has a longer break, consumed again DW. Those 8 people died because they stopped drinking DW, maybe once come back and drank it, but never after that. The average time after they finished the consumption of DW is 4.1 years. So the message is that if the DW consumption would be part of the oncotherapy, even in the remission, that would be a best way to prevent the relapse and the occurrence of the cancer. A couple of basic rules how to use it. Start the DW as close as possible to the diagnosis. Replace all the daily water intake with DW. Start with 105 or 85 ppm and reduce the deconcentration every 2-3 months with 20 ppm. The best is to drink as the patient get complete remission. If there is a complete remission, they can have a short break and start again, but do not forget to repeat the cure, the DW consumption.
And then people will ask about using DDW to reduce the incidence of cancer developing in the first place. But would you say that's a topic for another presentation?
Yeah, so we would say that people, a healthy population, they should consume 125 or 105 every year, every second year, every third year, depending on the risk. So the elder people should drink more frequently. The younger population, they can drink every third or fourth year, making a 3-4 months cure with DW. That would be good enough to reduce the incidence of cancer.
Okay, yeah, because in your book, it's the ranges between 105 and 125, and you've got people under 40 and over 40, and you've got groups of people who are healthy and have not smoked or they don't have obesity, and then you've got the group who have lived an unhealthy lifestyle. So they're the ones that would do it, I think you said, for 4 months at 105, and the other healthier population could do 3.5 months at about 125.
Yeah. This is what we suggest.
And also, what happens if people try and do things like drink 20 ppm for 4 months or 50? Is it just a waste of water, or does it have an undesirable result?
I have heard that somebody was consuming 10 ppm and they didn't feel well. So we don't know where is the limit. How fast should we reduce the D level to cause a very significant side effect or toxic effect? What we can say that the changing with 20 ppm, that's good enough to trigger the tumor necrosis and do not cause any side effect for the healthy cells and for the healthy population. I do not agree with the concept that the lower the D level, the better. I say that we have to optimize the deconcentration, so the whole population should reduce the 145, 150 ppm deconcentration to 130, 135 ppm. That would be the best. And drinking DDW with 125 ppm, 105, we can bring down the D level to this 130, 135 ppm.
Yeah, that's really important, because sometimes people assume, oh, doing it more aggressively must be better. So the take-home message is 30 years of your work and research shows that there's no need to make the deuterium concentration any lower than your studies. It's just not in line with the science and probably not necessary.
Yeah, I think so. It's a big game to changing and to determine the dosage depending on the cancer type and size of the tumor. So with a very aggressive tumor type, pancreatic cancer, glioblastoma, we immediately recommend the 85.
Mm-hmm.
And then we recommend a shorter period for one given deuterium concentration. And so and we need hundreds of experiment and new clinical study to find the best dosage. But in general, we can say I do not recommend to go immediately to the 25 ppm because that means that within 2-3 months will not be any changes in the body because reach the equilibrium and no other changing, lowering the level. So those cancer cells who survive that first 2 months, they slowly adapt to the low D concentration and they will start to grow again. So this is a way, and the aim to find real the best dose each type of cancer and for each combination.
Okay, that's so just sort of in simple language, you could make deuterium resistant cancer cell or deuterium depleted water resistant cancer cells. So I think that's just important for people who are listening, who are interested, that not to rush things and to follow what's been published already. Okay, we'll move into the next bit.
That should be the summary again. So in a healthy cells, the DH level is low, the cancer cells originally higher. So our conclusion is that on a submolecular level, we are able to intervene and modify all the process. So this is the 3 language which was published for Hungarian, English, Chinese, and Japanese. The English version available, and that was the reason I summarized all the knowledge we collected in the last 30 years, because of course it's very difficult to explain everybody one by one what would be the best way to use DW, but that is a help to everybody to get access to the basic information.
Yeah, so it's the Deuterium Depletion book it's on Amazon, and I've recommended it so many times, but there'll be the link in the description for people. And I think I've asked many times in this presentation about protocols because I know people will ask about it when they watch the video, and I think it's a very important book to read, and it's not difficult to read either. It's pretty straightforward, so I recommend the book a lot. Oh, so we've finished is there anybody you want to acknowledge from all of that data that you've showed already? Because we've got the next part of the presentation on blood sugar and diabetes.
I would say in that list of the people who took part in present that so many people helped that whole process and so many people had been involved to reach that whole science where we are today. And even there are much more scientists who did not collaborate with us, but they are also taking part in that journey. So I am very thankful everybody who helped that type of research in the last 30 years.
And it's a big list, and we've got USA, there's people in Hungary, we've got scientists in Iran. So it's basically a world effort, and it's a huge amount. So, I mean, the list here, I think there's probably 60 scientists, but you'd probably say maybe up to 100 were involved in where we are now with how important the studies are, showing the link between deuterium and cancer. And it's irrefutable, really.
Yeah. Yeah, so shall we continue with that surprise?
It's up to you. Yes, we can do.
Yeah.
I think some people are very interested in cancer, but also diabetes and high blood sugar is a big health risk. because it'll lead into cancer.
Yeah, so it was a big surprise for me that one day a patient with cancer and diabetes came to us, and he said that his blood sugar level decreased after starting drinking DW. And so I didn't believe that. Anyway, we went to the university, Semmelweis University, and we carried out 3 rat study. So in that study, we made the rats diabetic. And the next slide. In the very first study, the rat consumed 25 ppm and normal water, 150 ppm. And one group of the rats received insulin, the other did not receive insulin. And that slide shows that those group of rats drinking 25 ppm DW and receiving insulin, the blood sugar level was lower than the control. So we were very happy. And then we started another rat study. And in that study, the rats consumed tap water, 25, 75, 105, and 125 ppm. And the surprising result was that not the lowest D level reduced the best way the blood sugar level, but at 125 ppm, which is the closest to the 150 ppm. So we ran another rat study. Here we changed only with 5 ppm the concentration of the drinking water, and the next study, next slide shows the summary of the 3 experiment. It makes clear that the best dose to reduce the blood sugar level in the rat is 125 ppm and 140 ppm. And that was true for the glucose, and the next, that was true for the fructosamine, which shows the average of the last couple of weeks. And the next one.
Just for people who don't know what fructosamine is, what is it?
The fructosamine, and the next slide, the hemoglobin A1C, means the hemoglobin which is glycosylated. It means the more glucose concentration, higher the glucose in the blood, the more hemoglobin will be labeled with glucose. And that shows the average glucose concentration of the last 3 months of the patient. So if we can reduce the HbA1c, it means that the blood glucose level average is much lower at that range of the concentration drinking DW. So it makes clear that anyway, the DW reduce the blood sugar level, and all the hemoglobin A1c or fructose level shows that confirm that the data that the blood sugar level is lower. And the question is, what should be the mechanism? And in a normal case, the protein is called GLUT4, which is responsible to pick up the glucose from the blood. And the GLUT4 is staying in the cytosol. So when an insulin binding to the insulin receptor, that will trigger a signal system which will stimulate the translocation of the GLUT4 from the cytosol to the membrane. And that's responsible finally to take up the glucose. So in the experiment, we checked what is the GLUT4 concentration in the muscle membrane of the rat. The green column shows that a healthy non-diabetic rats, the GLUT4 level is high independently whether the rat consume 150 ppm or 25 ppm. But in the diabetic rats, the lowest was the GLUT4 level in the membrane, the rats consuming 150 ppm the concentration, the higher drinking 125 ppm. So that means that the rats at 125 ppm, the insulin signal system was stimulated and worked much more properly and could stimulate the GLUT4 translocation. So this is the conclusion of this rat study.
Okay, so that's really important, because sometimes if people read slides or listen to things in a hurry, they will hear deuterium depleted water and blood sugar and HbA1c going down, But the key in all of this is where it's dependent on whether insulin is present or not. So how would this transfer into people with high blood sugar?
Yeah, so it was proved in the next slide when we checked it in a human clinical study. But the next good news is that just the Tokyo University repeated our experiment with the rest in a tissue culture, and they could confirm it the same. So they could prove that the lower D level stimulate the GLUT4 translocation, which is very good for us that an independent laboratory confirmed our data. And nobody repeated our study with a human clinical study. So in that clinical study, the patients consume 104 ppm, 30 people were enrolled in the study, and everybody consumed it for 90 days. And we checked the deuterium level of the blood.
Because with these people, it said that they had altered glucose metabolism. So were they
were screened. They were screened for impaired fasting glucose level, diabetes, or impaired glucose tolerance. They were screened and they were enrolled into the clinical study.
Okay, so were some of them just insulin resistant, or would you say the majority really did have type 2 diabetes full blown?
Not each of them had diabetes. Some of them has a insulin resistance and impaired fasting glucose. So not all of them was at the late stage. I would say they got diagnosed with diabetes.
Okay, well, that's really important. So it's kind of now we're looking at ways that could prevent people going from going from insulin resistance into fully blown diabetic. So we're looking at people who are insulin resistant and
in the earlier stage. Yes. Yeah.
Well, this is really interesting now. So I'll let you carry on.
Yeah. So in that clinical study, we checked the level of the blood before the patient started to consume, and the average, if we go back, it was 147 ppm, and at the end of the clinical 133 ppm. So this is exactly the same range what we measured in the rat study. And the next slide shows the changes in the D level patient by patient. So at the beginning, there was only 4 ppm differences between the patients, 146 or 150 ppm. At the end, it was 125 and 143 ppm. And we checked several parameters of the blood. But for these parameters, we could prove significant differences comparing at the 0 and the days 90. The key message is that, for example, the serum natrium sodium level was significantly lower, which means that consumption of DW triggered the sodium hydrogen antiport, and the cells pick up the sodium from the blood, and that causes a decrease in the blood level, and that causes a decrease in the blood pressure. The so-called good cholesterol level, HDL, increased rapidly and very significantly. And the other very interesting point was that the white blood cell, red blood cell platelets, they increased. And that's good for the people receiving chemotherapy. We very frequently could prevent and prevent the blood system, and so they can tolerate much better the chemotherapy when drinking DW. The next slide shows that the fasting glucose level. So at the beginning, it was 6.07. At the end, it was 5.74. And everybody can see at 60 days, we had a peak. And the reason is that the patient's enrollment started in October. So 60 days later, that was Christmas time. and that disturbed our study. And I'm pretty sure that these numbers should have been much better if we started it in August, and the Christmas time do not disturb this clinical trial. We never do that again.
I was going to ask, in the study, the patients didn't change anything else about their lifestyle, such as diet?
No.
But this is a good example of just a week of eating badly or cheating can have a really horrible effect on blood sugar. And it took a while for it to go back down again, but we'll carry on. So I think that's an interesting observation. So the take-home message is that the blood sugar dropped after 90 days, but probably the peak at Christmas spoilt the data, and it would have been even lower if there hadn't have been the Christmas binging that happened.
Yeah. Yeah. And the other good news that so 30% of the patients we could see that reduce the insulin resistance. That means that the glucose uptake increased from 6.9 milligram per body weight per minute. It goes up to 8.64 milligram per body weight per minute, which is a good data. And I'm sure that if the study would last 6 months and not 3 months, not only 30% of the patients would reduce the insulin, much maybe all of them or almost all of them. So the preclinical study investigation proved that the DW decreased the serum glucose concentration, and the data of 30 enrolled patients in an open-label human study proved that significantly decreased the serum D level, increased the HDL concentration, and decreased the serum sodium concentration and blood pressure. This was
so
my
there's
conclusion.
also a hidden conclusion in this now, that it's reduced their cardiovascular disease risks now.
Yeah, I would say we need more study and more data, but in general, keeping the D level at 135, 130, 140 BPM, that will improve the whole metabolism, the whole body, and the people getting more healthy.
Yeah, definitely, because I know you're very scientific. That's why I pointed it out, because when I saw this data, that's the first thing that jumped out to me, was these are all markers for cardiovascular disease, except this particular study wasn't trying to prove that. And I think it's a really important thing for people to take on board who are interested in deuterium, or even people who are interested in taking it up as a research project, because we never know who listens or watches the videos.
Yeah. Yeah. Do we have another conclusion?
Yes.
Yeah, the glucose disposal significantly increased in 11 patients. It means we reduced the insulin resistance, and the study confirmed the pivotal role of nature occurring deuterium in the regulation of the physical processes. So that would be the end and the final conclusion. This belongs to the cancer study.
Okay.
And
that's
yeah.
fine. Oh no, that's so basically the last studies that we've looked at exploring insulin resistance, high blood sugar, this is sort of a new, are you going to do more studies in this area? Because where are you going next with your studies? More cancer patients, or are you going to do the diabetes and blood sugar route?
And both. It depends on the funding. So yes, we already have a protocol to start a study with a diabetic population, and I hope we can start it soon. And of course, we have to complete new clinical study with the cancer patients to get the approval at the FDA or the EMA. Both indication is very, very important for the whole society, because if we can solve that two problems, that should be much better for the health.
Oh yeah, no.
I mean, for the society.
Yeah, that was absolutely fascinating. Thank you for sharing that. And I think people will understand the data because it's very powerful, and it's backed by so many other scientists, and there's so many publications over the last 30 years. Is there anything else you wanted to mention on the subject of deuterium, or anything in the presentation that you think is important?
I think it's important that our company, the HYD LLC, is in a part of a consortium that, oh, good for you.
Yeah.
I got that one.
Yeah.
So the European Union is supporting a study, 0.6 million euros, a consortium involving the university from Portuguese, Lisbon, in Würzburg University and the Oxford University. And that consortium is prepared to test the effect of deuterium on the nervous system. And hopefully the results and the experiment has been done, and the results will come soon. And we could, and of course, here again, they could see a big effect when we modified and reduced the deconcentration. So, and this is on the way, and the research has started not only the cancer or metabolic disease, but the central nervous system related diseases. And I hope the scientists summarize the data and they will publish it soon.
Which central nervous system diseases in particular were they looking at?
In that study, they treated the brain, one half of the brain, they killed. And the question was whether the deuterium depletion, how can modify and protect that type of effect. And the conclusion will be that somehow the lower D level can prevent that area of the nervous system. That will be one conclusion. I hope they will come out with these type of results.
All right, so it's kind of in terms of lay people listening, it would be imagine if somebody'd had a brain injury, because I was thinking of things like Parkinson's and multiple sclerosis and DDW, but from what you described, if they're cutting out half the brain, it's looking at a brain injury and how can deuterium depleted water change the outcome.
This is one of the focus which can count, yeah.
Okay, and what about, because people ask me about things like multiple sclerosis and Alzheimer's and Parkinson's and deuterium. Would you say the studies are coming, or do you know of any?
In general, I would say that the lower D level must improve the efficacy of mitochondria, and if the mitochondria work more properly, that can make the central nervous system stronger, and that way maybe can prevent, or we do not have any data with that type of disease, Alzheimer's or any, but in general, if that type of positive data come out, I can explain with that results that somehow helping the mitochondria better metabolism can prevent that type of disease.
Okay.
Appear.
So the take home from that is if somebody does have a neurological situation, they can use deuterium depleted water safely to improve their mitochondria, but at the moment there aren't any definitive studies to say that it will help their particular neurological situation, but it will help their mitochondria and very likely improve their quality of life, but it's not claiming to cure any kind of neurological situation.
Yeah, so we do not have any data to confirm that directly.
Okay, great. Anyway, thank you very much for the treat of biochemistry and talking about all of the studies. I really appreciate it, and I'm sure people will find it really interesting.
Thank you, Sara. Thank you.
Thank you very much for watching. I hope you found this useful, and feel free to share it with anybody you think will benefit. And this particular episode is going to lead in very nicely to Dr. Borosh, who's going to talk about the new deuterium research that's just come out, because Gabor's given us a really good overview of the last 30 years and his work. Thank you for